giving me the opportunity to present here today. So coming back to the first question that was actually asked, so in our work, we focus on reconstructing phylogenetic trees using using SMVs. So we do use point mutations. So the clonal theory of cancer states that a tumor, is the result of an evolutionary process. And this process starts at a normal cell, which acquires a mutation that gives it a selective advantage. And this in turn leads to an increased replication rate. And the descendants of this normal cell may acquire new mutations themselves too, leading to further selective advantages. And this results in the formation of clones, which are collections of cells with identical mutations. We can represent this process as a tree. It's a rooted tree. And the vertices of this tree correspond to the clones. And edges encode direct parental relationships. And they are labeled by the mutation that distinguishes the parent from the child. So when we look at a tumor at the present time, we actually have a heterogeneous mixture of cells. So this is known as intratumor heterogeneity. And from this heterogeneous mixture of cells, we actually want to infer the tree. And the reason why we want to do this is because it has applications in personalized cancer medicine. So for instance, if you would know the tree, you could target subclonal mutations that confer resistance or that have metastatic potential. So let's take a closer look at this heterogeneous mixture of cells. And the goal is to infer this tree, and we do that by sequencing. So in cancer, there are many types of mutations. And here we focus on single nucleotide variations. In that case, our data corresponds to variant allele frequencies. And these are fractions of variant reads at a position. So in this case, we have 5 reads, 2 of which have the variant allele, and this corresponds to a variant allele frequency of 0.4. So for SMVs, it's reasonable to assume that a mutation only occurs once throughout the entire history of a tumor. This is the infinite sites assumption. And in addition, to only restrict to SMVs that are unaffected by copy number aberrations. So under these two assumptions, the variant allele frequency of a mutation is proportional to the number of cells that have the mutation. And this is a great result that we actually use throughout the rest of this talk. So let's zoom in a little bit on this sequencing. So as mentioned by the previous speaker, if you would use single-cell sequencing, we wouldn't have much of a problem because basically our data would correspond to measurements of individual cells, and these are directly the vertices of this tree. So there's no mixing going on. And because of the infinite sites assumption, we can apply perfect phylogeny theory to infer this tree in linear time. So that's if you would use single-cell sequencing. But unfortunately, most of the data is obtained by bulk sequencing. And in bulk sequencing, your samples correspond to heterogeneous mixtures of cells. And this in turn corresponds to a heterogeneous mixture of vertices of this tree. So we're given variant allele frequencies, and we want to find a clonal tree T. So recently, tumors have been sequenced at multiple sites. And this results in a variant allele frequency matrix where the rows correspond to the different samples. So given this frequency matrix, we want to infer the tree. And we also want to infer the mixing proportions for each of the samples of the tumor. So the whole problem basically is, given this frequency matrix, find the tree and find the mixing proportions that describe the composition of each sample such that you actually have this frequency matrix. And since this is a heterogeneous mixture of cells, you cannot apply perfect phylogeny theory immediately, and you have to think about how to deal with the mixing. So when we started this project, we had 2 fundamental questions. So the first question is a question about the existence of a tree. So given an arbitrary frequency matrix, can I find a tree that I can mix to obtain the frequency matrix? So that was our first question. And the second question is about finding conditions that we can use to characterize the whole set of trees that are compatible with a frequency matrix. So in order to answer these 2 questions, we assume error-free data because we don't want to deal with noisy measurements. We just want to focus on the combinatorial structure of this problem. So this is an assumption that I will make in the first part of the talk. And under this assumption, I will formulate a problem statement that formalizes the notion that I just presented in the previous slide. I will show necessary and sufficient conditions to solutions of this problem. I will say something about the hardness of the problem, and I will give an algorithm for solving the problem. Next, I will show how these results extend to noisy measurements by introducing a probabilistic model, and I will conclude by showing results on simulated data and on real data. So let's formalize the problem. So we're given this frequency matrix F, and we want to find this tree T. So the first thing we do, oh, this is unfortunate. So the first thing we do is we use perfect phylogeny theory, I guess you can see it over here, to encode this tree as a binary matrix. So the rows of this matrix correspond to the vertices of the tree, and the columns, they correspond to mutations, and there's a 1 if the clone has the mutation, so if the vertex has the mutation. And what we in fact do is we restrict the set of matrices and trees by requiring that every edge is labeled by exactly 1 mutation. So this is a stronger assumption than what you'd normally have in perfect phylogeny theory. And when making this assumption, we can actually show there is a 1-to-1 correspondence. So we prove this between the set of restricted binary matrices and the set of allowed clonal trees. So you can use these two interchangeably. They correspond to the same thing. So we have this frequency matrix, and we have this binary matrix, and we need something in the middle to explain the mixing. And this is the usage matrix. So the rows of this matrix correspond to the samples, and the columns, they correspond to the vertices of the tree. And since these are mixing fractions, they have to be non-negative. And in addition, since they describe an entire sample, they have to sum up to at most 1. So these are the two conditions that we impose on the usage matrix. Now the question is, we have these 3 matrices. What's the relationship between those matrices? It turns out that the frequency matrix, F, is 1/2 times U times B. The reason why we have 1/2 is because we only focus on heterozygous mutations. And this is a nice linear algebra relation. So to summarize, we're given this frequency matrix F. And basically what we want to do is we want to factorize this matrix into 2 matrices, U and B, whose product times 1/2 equals the frequency matrix. So this is a kind of a non-negative matrix factorization problem with additional restrictions on the 2 matrices. So many authors have studied different variants of this problem in both the single sample case and the multi-sample case. And in our work, we were really interested in the fundamental question about solutions to this problem. So what are the conditions that describe solutions to this problem? And let me start by focusing on the first question that we had. So if I'm given a frequency matrix F and a tree B, can I find a usage matrix that I can plug into this formula? So is there only 1, or are there many? So that was a question that we initially had. And we answered that question by taking a closer look at the binary matrix B. And it turns out that this is a very strong matrix. So it's a square matrix. And in addition, it's also invertible. And because of that, we can basically use linear algebra to describe U in terms of F and B uniquely. So that answers the first question. So given F and B, there's only 1 usage matrix, or 1 matrix U. So we don't know if it's a usage matrix yet. So instead of computing the inverse of B, we have a non-recursive closed form equation of U in terms of F and the tree, matrix B. And I'd like to point out that different versions of this formula have appeared in previous work, but they've been defined in a recursive way. And I will show you later why it's better to actually do it non-recursively. So here's an example about this matrix U. So here I'm using an F and a B, and you can see that there is a negative entry in the matrix U. So this is not a usage matrix, but this is the only matrix that you can plug into here. So the question is, when does this actually happen? So why do we have a negative entry here? And can I describe conditions that tell me that when they are violated, that you will get a negative entry? So that's the thing that I will do next. I will define 2 conditions, the ancestry condition and the sum condition. And let me start by describing these 2 conditions. So here I'm showing you a tree, and I've plotted the frequencies of the first sample on the nodes of the tree. So the ancestry condition states that basically every branch in this tree has a decreasing sequence of frequencies. And you can see that it's indeed the case. So that's the ancestry condition. And the sum condition states that if I look at a parent, the sum of the frequencies of its children should be at most the frequency of its parent. So these two conditions are necessary. And by that I mean that if I have a solution to the VFP, so given a frequency matrix F, a tree B, and a usage matrix that I can plug in and is actually valid, so non-negative entries, then these two conditions have to be met. So that's what I mean by necessary. And it's actually easy to see why they are necessary, and it follows immediately from this non-recursive definition of the usage. So for instance, over here, for the usage to be non-negative, the frequency has to be bigger than the sum of the frequencies of the children, and that's exactly this condition. So it turns out that the sum condition is also sufficient, and by sufficient I mean that if I'm given a tree that satisfies the sum condition, then I also know that the corresponding usage matrix will be valid. It won't have any negative entries. So the sum condition provides a complete characterization of the set of solutions to a VFP instance. So if I go back to this example where we had a negative entry, since the sum condition is necessary and sufficient, it must be violated in this case, and that's indeed happening. So 0.15 is smaller than 0.16. So we have a complete characterization now of the set of allowed solutions, answering the second question that I had up initially. And now the question is, can we somehow capture this combinatorial characterization, or can we somehow capture this complete characterization into a combinatorial structure? And that's what I will show you next. I do that by defining an ancestry graph. The vertices of this graph correspond to mutations, and there's an edge between two mutations if they satisfy the ancestry condition. And the observation here is that a spanning tree corresponds to a solution, or may correspond to a solution. And this is also an observation that was made recently in work by Popik et al. And here we prove that this is a correct observation. So stated more formally, you have a clonal tree T that is a solution to a VFP instance if and only if the same tree is a spanning tree of G that satisfies the sum condition. So I forgot to mention this. So this tree over here has to satisfy the sum condition. It's not just any tree. So finding a spanning tree in a graph is easy. You can just do that by using DFS. But finding a spanning tree that satisfies the sum condition is hard. So we show this by reducing another NP-complete problem to the VFP. So it's not easy to do this. That's the conclusion here. Good, so we have a complete characterization of solutions, and we know it's hard. So how do we find solutions? So we do this using an ILP formulation. I'm not going to go through the details. The only thing I'd like to point out is the objective function. So we try to find the largest tree that satisfies the sum condition. And if this tree spans all the vertices, only in that case you have a solution to the VFP. That's basically what's happening here. So to summarize, I've started by focusing on error-free variant allele frequencies. And I've introduced the variant allele frequency factorization problem. I've shown necessary and sufficient conditions to solutions of that problem. I've mentioned that the problem is NP-complete without showing a proof. Proof is in a paper, by the way, in the supplementary material. And I've described an algorithm for solving this problem. So if this were a math talk, I'd be done. But in CombBio, we have to deal with noisy measurements. So that's what I will do next. I will show you how we do this by introducing a probabilistic model. So let me just go back to the input frequencies that we had. So a variant allele frequency is defined by the number of variant reads divided by the total number of reads. And in our model, we look at the reads as draws from a binomial. So more specifically, the number of variant reads is a binomial parameterized by the total number of reads and the true variant allele frequency. Now, if you turn this around, you can ask yourself, what is the posterior probability of having a VAF given the read counts? And this is actually a beta distribution. So what we have instead is we have a matrix of beta distributions, one for each mutation per sample. So basically, matrix of frequencies becomes a matrix of distributions. And the cool thing is that the means of these distributions actually correspond to these frequencies over here. So just to go back to what we did previously, so we defined the ancestry condition by comparing frequencies in this matrix. So essentially what we were doing is we were basically looking at point estimates and saying that a mutation, the purple mutation, is ancestral to the green mutation, only if the frequency of the purple mutation is at least the frequency of the green mutation. So we can do the same thing using the distributions. So, and we do that by defining a posterior ancestral probability that the purple distribution is greater than the green distribution. So we get basically probabilities now instead of just what we did over here. So these are posterior ancestral probabilities, and we can actually compute these posterior ancestral probabilities analytically, since they are beta distributions, since the underlying distributions are beta distributions. So let me show you how we actually use these posterior distributions. So there are two possibilities. So if I look at two distributions, they could overlap, such as here, or they could be really separate. So in this case, there's a clear ancestral signal. And over here, we cannot really distinguish between the two mutations because we don't have enough power. So what we do is we define an approximate ancestry graph, and the vertices of this graph correspond to sets of mutations that we cannot distinguish, whose distributions overlap. And the edges, they correspond to pairs of mutations, two mutations, where you have a clear ancestral signal. So this is the approximate ancestry graph. And let me just quickly illustrate this on a real tumor. So this is patient RK26. It's a clear cell renal carcinoma described by Gallinger. And what I'm showing you here is the approximate ancestry graph. And you can indeed see that vertices correspond to sets of mutations. So if you, for instance, zoom in on this one, this is the picture that you could have. So you have a bunch of overlapping distributions for each of the mutations that you cannot distinguish. That's why they end up in this cluster. And the edges, they correspond to they connect vertices that have high ancestral probability. So this is the graph that we use to search for, so we use this graph to search for a solution. And in our case, a solution is the largest tree that satisfies a relaxed sum condition. So the steps that we take in Ancestry, our algorithm, is we build this approximate ancestry graph, and then we find the largest tree that satisfies a relaxed sum condition. And we do this using an ILP. And once we have this tree, we also compute a usage matrix using the equation that I just described to you. So this brings me to the final part of my talk. I will show you results on simulated data and on real data. So let's start with simulated data. So we compared Ancestry to 3 recent methods: PhyloSub, SITUp, and Litchi. So these methods deal with multi-sample data, and they also infer a tree. And the way we set up our simulations is by simulating a tree at random. So basically, we generate a tree randomly, and we mix the vertices of this tree also randomly. And then we infer read counts and also variant allele frequencies. So, and this is actually the input that we give to each of these methods, including Ancestry. And then we compare the inferred trees to the true trees. And we do this using 3 metrics. So the first metric I will show you looks at pairs of mutations that occur in the same branch. So I call that ancestral here. And what I'm showing you in this violin plot is the accuracy. So what is the fraction of mutations that are truly ancestral, that were correctly identified as being ancestral in a inferred tree? And here I'm showing you the accuracy values across all 90 instances. And you can see that Ancestry outperforms the other methods. So these are the median values. We also looked at other tree measures. So we looked at the fraction of correctly identified clustered mutations. That's this violin plot over here. And the fraction of correctly identified mutations that occur in different branches. And in both measures, we also outperformed the other methods. So there's one thing I'd like to point out. So both Ancestry and Litchi do not use all mutations. So what we actually did in these violin plots, we restricted them to the set of mutations that were inferred by all methods. And in our case, we don't use all mutations because we only focus on those mutations for which we have high confidence. So this brings me to the next slide. This is a CLL patient, a leukemia patient. And it's been sequenced at extremely high coverage. So there was only a panel of 16 genes, 16 loci that were sequenced. And the coverage is 150K. And what I'm showing you here on the left is the tree inferred by Ancestry. And here on the right, I'm showing you the trees reported by PhyloSub and SITUp in their respective publications. And one thing I'd like to point out is that, for instance, if we focus on these 2 mutations, we separate them out, And if you look at the posterior distributions, we can really see that that they are really separate if you look at the 99.99% confidence intervals. And we also infer the usage matrix. There's one caveat I'd like to make. So our reads are actually affected by PCR, and this is something that we try to incorporate into our model as well. So let me just conclude quickly. So in summary, I've described, I focused on the error-free case first. I formalized the problem that we call the variant allele frequency factorization problem. I've shown necessary and sufficient conditions. I've shown that the problem is NP-complete, and I've described an algorithm. Next, I've shown how these results generalize to noisy measurements by defining a probabilistic model. And the resulting algorithm reconstructs clonal trees accurately. We've shown this on simulated data. And it's also able to exploit higher coverage data to identify successive clonal expansions. So this brings me to my final slide. I'd like to acknowledge my collaborators. So Leila Usper, I share first authorship with her, and Hannah and Ben Raphael. And finally, I'd also like to acknowledge the organizers for awarding me a travel grant. Thanks.
Thank you. We have time only for one quick question. Well, maybe two if we're very quick. Let's start here. Saurabh. Sorry.
Okay, so very nice work. I'm quite concerned about the assumptions and how they generalize to the majority of tumor types that will exhibit significant copy number alterations across the genome, and so your assumptions won't hold in the majority of cases. So I'm just wondering what your thoughts are on that.
Yes, that's a great question. So the reason why we focus only on regions that are unaffected by copy number aberrations is because then you have this really nice relationship between the variant allele frequency and the proportion of cells that have the mutation. And by making this assumption, we could actually really characterize the combinatorial structure of the problem. And we are now currently looking into ways of incorporating copy number aberrations, and this work really helps us a lot because we know what's going on with the mixing.
I had the same question, so it was answered.
Oh, okay, great.
Maybe I have the same question or different, I don't know. The way I ask you is how, before you reconstruct a tree,
yes,
how do you know that there is a tree? So what if there are many trees or they're all independent?
Yeah.
Because you basically play with the timing of the mutation. That's the only thing you know.
So the question is, how do you know if there is a tree?
Yeah.
So actually I can construct a matrix, frequency matrix, that does not admit a tree. So that can actually happen if you have error-free data. And then you have the component of noisy measurements. And we try to deal with this noise by looking at distributions. So instead of trying to find a tree that spans all the mutations, we find a tree that explains the largest set of mutations. So that's how we deal with it. We don't try to basically have a tree that really explains everything. Thanks.
Oh, great. Okay, let's thank Mohammed again. And we move on.